Right. Hello, everybody. My name's Aditya. So over the past year, I've been working with my BME group on targeting neurons, or targeting DNA nanostructures into neurons. So just a brief overview of why we do this. So the World Health Organization reports that approximately 6.3% of the global burden of disease can be attributed to neurological diseases, and that this further accounts for 12% of all deaths globally. And there have been some significant trends with regards to this. Number one is that this is not just a problem of the rich countries anymore. Poor and rich countries, developing countries, resource-constrained societies, they're all facing neurological disorders. And the second thing is that this is not just restricted to psychiatric disorders like dementia or Parkinson's. These are fairly neurosurgical, like biological disorders such as glioblastomas, astrocytomas, brain tumors, etc. So there's two main problems in trying to study and target therapies to these brain disorders. First is the blood-brain barrier. So this is basically a membrane that exists to protect your brain from toxins that exist in your bloodstream. But it also poses a problem for therapy because drugs that are injected into the bloodstream to target the brain do not cross over in adequate amounts. The second problem is actually targeting neurons within the brain itself. So the brain is actually 10% neurons, the cells that actually do the brain's tasks, such as thinking, making decisions, etc., and 90% glial cells, which are just supportive cells that nourish neurons. There are certain disorders that are restricted just to glial cells, like glioblastomas, and there are certain disorders that are restricted just to neurons, like Parkinson's. So we want to be able to target our therapies specifically to the kinds of cells that are affected. And so we need a carrier for these drug molecules or diagnostic devices. And so the design that we focused on was based on DNA origami. I'm not going to go into details of this, but it's basically what it sounds like, applying a traditional Eastern form of art to 21st century technology. So it's basically folding of DNA at the nano level. And so you can see on the images on the right that from 2006 onwards, groups have come up with various shapes of DNA at basically the nanoscale, and you can make any arbitrary two-dimensional and three-dimensional shape. So the design that we focused on was this 20 by 30 by 40 box, and it's ideal for our purposes because it folds at the nanoscale at a high level. It's durable. Although we have not tested it yet, reports have come in that it's stable in the bloodstream, and the bloodstream can be a pretty violent place for molecules so small. And it's ideal for our purposes because it has a cavity in the middle, and so we can use it to hold cargo if necessary, using chemical binding, of course. But I want to emphasize that this DNA nanostructure does not have any intrinsic biological properties. By itself, it will not do anything. So we have to basically give it some biological properties. And so the way we do it is we take the standard box and we add what are called oligonucleotide extensions to the sides, which you can think of them as sticky ends. They're basically single-stranded DNA. And so if you have two sides of a Velcro, for example, and you just have one side sticking out, that's basically what it does. And so once we do that, we're going to functionalize them with our peptide of interest, which is a protein derived from the tetanus neurotoxin. So as you may know, tetanus affects the motor system. So the tetanus neurotoxin gets into motor neurons, and that's how it has a specific cellular mechanism of transcytosing past the cell membrane. And so once we add those peptides onto our box, that will conclude the functionalization process and give it the power needed to get into specific neurons. And then we go on with animal testing. So specific aims really quickly. We take our literature DNA box and sort of add the designs that we intend. We create the polymer cap, which consists of the peptide, attach it to the box, and test if it forms properly. And finally, we go ahead with in vitro and in vivo testing. So real quick, how do we actually do the design? So on the left, bottom left, you can see the original literature box as is represented in a software called CAD Nano, which allows for these arbitrary three-dimensional DNA nanostructures to be created. And what our innovation was taking basically these extensions, orange extensions, and extending them past the face of the box. And those basically constitute the sticky ends that I talked about earlier. And so Each of those sticky ends were actually 14 base pairs long, 14 thymines long, and just to allow for like non-probabilistic binding and things like that. And so we added a staple strands and our actual scaffold strands, which basically form the entire box. And the beauty of this is that once you add all the reagents together and just heat it up, it forms automatically at the nanoscale. So the hard part was done for us. And so these are our preliminary results. You can see on the right that our structures formed in a relatively high yield. Like each of these like vaguely represents one of our boxes, and you can sort of see like the cavities in the middle at this resolution. What you can't see at this resolution are the sticky ends that come out of the boxes, but they are there. They have been like verified by testing. And the gel on the left just shows a screen of like magnesium concentrations that we ran to determine the optimal folding concentrations, because we want our yield to be as high as possible. Next, we wanted to determine if what we formed was actually functional, if it was actually working. So we did another gel to quantify the fluorescence of the box, because we need a way to like actually see that the box is getting into neurons in the brain. So what we did was first try to see, we loaded 4 wells in the gel. The first was just a regular box. The second was the box with the sticky ends. The third was regular box with a fluorophore floating around, and finally was our box of interest, was the box with the fluorophores attached. So the first thing, gel on the right, we wanted to see exactly where the fluorescence was, and what we saw was what we expected. The fluorescence was localized only to the last lane. Secondly, we repeated the gel with the gel dye, and we saw what all the relative distances were. And finally, once you co-localize these two images, you can see that the fluorescence was restricted only to the last lane and traveled the expected distance, thus confirming that our peptide cap was able to attach to our DNA structure. And finally, we went on with 2 phases of testing. The first was in vitro testing, where we took our nanostructures and applied them directly to a plate of neurons. And then we imaged those cells to see if the fluorescence was retained. Secondly, we did animal testing to see if so we injected these neurons into the tail of a mouse, and then the brain was later extracted, and we imaged the brain to see if the neurons were able to enter like specific cells in the brain. So results of our preliminary cell internalization experiments. So these images, the one on the left, the one on the left is the bright field image, and the one on the right is the fluorescent image of the same. And so this background is actually just a sheet of glial cells. And the preliminary results are promising because we are able to identify specific neurons within the sheet of glial cells. So remember I said earlier that brain is 10% neurons, 90% glial cells. You can kind of think of the same proportions here. 90% of the cells here are glial cells, 10% are neurons, and approximately 10% of the cells have been identified. So basically, the cells stained in red are neurons. And on the bright field, you can sort of see that similar effects are going on, although some of the cells have died. A minor setback was that some of the cells were killed by the folding buffer we used to contain the neurons themselves. And so that might have induced some sort of like cellular entry independent of our tetanus neurotoxin. So we're conducting experiments to see the validity of that. And we also tried changing the buffer, but that did not improve the results. So we might consider like modifications to our design. But these experiments were successful from a design perspective. We were able to create like numerous variations of the sticky ends that I was talking about, 14 thymines, 15 random sequences, things like that. We were able to functionalize our DNA nanostructure, give it some sort of biological power, and we were able to create a polymer cap and be able to like specifically attach and test the attachment of that to our boxes. So future work might involve changing the structure of our polymer cap. So we used a 10,000 molecular weight PEG molecule as a spacer molecule between the sticky ends and our peptide of interest. So we might just change that to a molecule, just basically this guy here. So the sticky end would bind here and the peptide would bind here. And it's a much smaller spacer molecule, giving us much more control. So we hope to test that. And our initial in vivo experiments were somewhat promising, but it still needs a little bit more work about quantifying the amount of cells that are actually identified with these nanostructures. Finally, I want to thank my advisor, Dr. Coleman, for his support throughout the years, and the Han Lab, and of course, Kiloton Honors College. And I'll be happy to take any questions if we have them. Yeah.
In an in vitro setting, is there a way to move the boxes somehow so you can get them closer to neurons or move them in space?
What do you mean move them
Like
closer?
applying a gradient of some sort?
Like you mean just the sheet of cells?
Yeah.
We haven't tried that. So like yeah, sure. So the question was there a way to like move the neurons together basically during in vitro testing? And so we haven't tried that yet. So the cells we get are just a plate of cells, and then what we do is we add our structures, and then we wash the structures off, and then we wait about half an hour for the reactions to take place, and then we just go ahead and image it. But yeah, I mean, so like after our initial cell testing, after we changed the buffer, we haven't been seeing like that promising of results. So that's why we're considering changing the design of the polymer cap to see if like the cells take up these structures with more specificity. Yeah.
What kinds of things like would you put in the box to deliver to the cells? Like,
All right. So the question was what kind of cargoes the box could carry, basically. So there's two main applications. There's diagnostics and there's treatment. And so for diagnostics involves like imaging, basically, right, and identifying like certain causes of diseases. So what you could do is maybe you could add like some kind of, let's say a quantum dot, right, into the box, or like a more like bioactive fluorophore for imaging purposes. Or for treatment, you could do a bunch of things. So groups have done experiments where they intercalate chemotherapy drugs into these DNA nanostructures and use those to like bring them into cells, drug delivery applications. And then basically, let's say you have someone who has a hormone receptor deficiency, right? Like you don't want to like get into that problem, so you just bypass the whole problem by adding your hormone of interest into the cavity and bringing that into cells. So the applications are limitless, but we're still in the preliminary phases of like figuring out the details of how we're going to get the box ready and things. All right, thank you.